Hello everyone, this is Dr. Komalacharya and here I am presenting the new video of this video lecture series, and the topic is gene transcription in prokaryotes. If you remember, in my last video we learned the basic idea of the central dogma. We saw that the information stored in the DNA is used both in the DNA replication for cell division and in the gene expression. Now what the gene expression is? Gene expression is a process by which the information stored in the DNA is converted into functional products like a protein. But a very first step of the gene expression is the gene transcription. Transcription is basically a process of RNA synthesis, in which a cell makes a copy or a transcript of a piece or segment of a DNA that is known as the transcript is known as RNA. Here you can see the basic structure of RNA. RNA is ribonucleic acid, which is composed of the pentose sugar, phosphate group along with the nitrogenous base. A nitrogen base could be cytosine, guanine, adenine or uracil. In case of DNA, it's thymine. Here in case of RNA, the uracil replaces the thymine. Adenine is complementary to the thymine in case of the DNA, whereas in case of RNA, adenine is complementary to the uracil nitrogenous base. These are different types of RNA: mRNA, rRNA and tRNA. Each of these RNA has different functions. Like mRNA carries the coding information for the protein synthesis, rRNA are the components of the ribosome, whereas tRNA carries the amino acid during the translation. So this is the very basic information regarding the RNA. We will talk about all these things in detail in my next video lecture. But now let's focus on the transcription. So as I told you earlier, transcription is a process of RNA synthesis. But for transcription, a cell requires three basic things: transcription proteins or enzymes. Basically, RNA polymerase enzyme carry out the whole transcription process. Second one is ribonucleotides, and third one is template strand. Now as far as the template strand is concerned, we all know that in replication whole DNA molecule is replicated, but it's not like that in case of transcription. Transcription is a selective process in which only a particular segment of genome is transcribed into RNA, and this part of the genome is known as the transcription unit. So transcription unit is a segment of DNA that is to be transcribed into RNA. A transcription unit has transcription start site, from where the transcription begins, and the transcription termination site, where the transcription ends. Now there are two new terms, upstream and downstream. The sequence prior to the transcription start site is known as the upstream region, and the sequence after the start site is known as downstream region. Downstream region contains coding information, while the upstream region contains the promoter region. Before going into detail about the promoter, let me tell you one more thing. Unlike replication, in case of transcription, only one strand is used as the template strand to guide the RNA synthesis. The template strand is always oriented in 3' to 5' direction. And another strand, which is complementary, is known as a non-template strand or coding strand, that is identical to the RNA transcript that is to be transcribed, and it is complementary to the template strand. Okay, so this was the transcription unit. Now let's talk about the promoter region. Promoter region is a DNA sequence where the RNA polymerase binds first. It's a DNA sequence that serves as the initiation signal for transcription process, and it's a site where the RNA polymerase is attached initially. Now promoter contains two consensus sequences, -10 consensus sequence, that is also known as Bribnow box, and -35 consensus sequence. Both of these consensus sequences possess 6 nitrogen base pairs. Now the question may have arisen that what is this -10 and -35? So the first nucleotide at the start site, which serves as template, that is designated as +1, and the nucleotides downstream from this spot has given positive values, whereas the nucleotides upstream of this spot is designated as is given negative values. So the -35 and -10 region, -10 shows the approximate distances in terms of nucleotides from this start point. So this was the promoter region. And this Pribnow box, or we can say -10 sequence, contains TAT-AAT sequence nitrogen base pairs, whereas the -35 consensus sequence possesses TTGACA nitrogen base pairs. Okay, so the promoter region is where the RNA polymerase binds initially, and now let's talk about the RNA polymerase enzyme. It is DNA dependent enzyme that catalyzes the synthesis of RNA from DNA. Actually, the RNA polymerase holoenzyme is composed of two components: RNA polymerase core enzyme and the sigma factor. RNA polymerase core enzyme is made up of 4 different subunits: alpha, beta, beta dash and omega. Basically, the core enzyme is made up of 2 alpha subunits, 1 beta dash and 1 omega subunit. This core enzyme is non-functional. It becomes active only when it binds with the sigma factor. The sigma factor does not play any role in catalytic activity, but it has the capacity to recognize the promoter region. Okay, so each of this subunit has different functions. Like alpha subunit play role in assembly of core enzyme and promoter recognition. It also interacts with some regulatory factors. Whereas beta and beta dash both together make the catalytic core center, and beta subunit is also involved in the chain elongation process, while the omega subunit facilitates the assembly of RNA polymerase holoenzyme. It also stabilizes the assembled holoenzyme. And as I told you earlier, the sigma factor helps in the promoter recognition. So both these core enzyme and sigma factor together makes RNA polymerase holoenzyme. It is the largest bacterial enzyme. Now, as far as the transcription process is concerned, it can be divided in 3 different steps: initiation, elongation and termination. Let's begin with the initiation process. Transcription begins when the holoenzyme binds to the promoter at -35 consensus sequence and form a closed binary complex. Here, closed binary complex suggests that the DNA remains in duplex stage. Okay, so DNA is in double helical structure, and only the holoenzyme can initiate this process because the core enzyme cannot differentiate between promoter and other sequences. Sigma factor recognizes the consensus sequence and alter the binding property of RNA polymerase, and so that it can bind with the promoter region. After the formation of the closed binary complex, the holoenzyme shifts to the -10 consensus sequence. Now -10 site is AT-rich region, and that makes the breakdowns of the bond between the two strands of the DNA more easier. So after the formation of closed binary complex, melting of the short DNA segment that is already bound to the enzyme starts, and the double helical structure of DNA opens up and form an open complex. Here you can see the open binary complex. That is followed by the unwinding of other 12 to 14 base pairs, and as a result a transcription bubble is formed which contains a temporary DNA-RNA hybrid. After that, incorporation of new nucleotide begins using a template strand. First nucleotides added are usually purines. And now the phosphodiester bond between ribonucleotides are formed, and as a result ternary complex is formed that contains enzyme, DNA and RNA. Generally, after the addition of 8 to 9 nucleotides, sigma factor dissociates from the core enzyme and the core enzyme takes over the further process. The enzyme remains at the promoter region until it incorporates around 9 nucleotides. After addition of 9 nucleotides, the enzyme moves on further. On on the template DNA strand. Now let's talk about the elongation step. During the elongation process, the transcription bubble along with the RNA polymerase enzyme moves on further. As the enzyme moves, it unwinds the DNA helix to expose the new single-stranded segment of the template. Here you can see the elongation site. Right? Now, what happens during elongation is the new ribonucleotides are added to the 3' end of the growing RNA chain. Here you can see the DNA template strand, and this is the RNA growing chain. So the new ribonucleotide is added at the 3' end of the RNA growing chain. And meanwhile, 2 phosphates are released as pyrophosphates, okay, which will break down further, which will hydrolyze further, and it provide energy for this whole process. Apart from that, one hydroxyl group is also removed from the 3' carbon of the nucleotide that is already present at the growing RNA chain. And this is how the nascent RNA chain is synthesized in 5' to 3' direction. As the elongation continues, 2 strands behind the transcription bubbles resume their double helical structure. So, during this whole process of elongation, RNA polymerase unwinds and rewinds the DNA helix. So, as the RNA polymerase moves forward along the DNA helix, it generates positive supercoiling ahead and negative supercoiling behind. So, to complete this elongation process successfully, we need some mechanism. So here the gyrase and topoisomerase 1 enzymes participate. Gyrase enzyme removes positive supercoiling by introducing negative supercoiling, whereas the topoisomerase 1 introduce positive supercoiling and release the tension of negative supercoilings. So this was the elongation process. Now let's move on to the termination step. When the RNA polymerase reaches to the DNA sequence that serves as the terminator, it stops adding new nucleotides and the transcription process terminates. But how exactly this occurs? Bacteria use 2 different strategies for this termination. One is row-dependent termination and another one is row-independent, or we can say intrinsic termination. Let's talk about row-independent termination first. Here the intrinsic terminator means the sequence, the DNA sequence that gives the signal for the termination process. Okay, so in this case of termination, the terminator has GC rich inverted repeats. So the RNA transcript that is synthesized will have the abundance of GC base pairs, and this results in the formation of hairpin loop-like structure in the transcript. This hairpin loop-like structure is followed by uracil-rich region, and this poly-U sequence causes the RNA polymerase to pause for some time. Besides that, the AU base pair here is too weak to hold the DNA-RNA hybrid together, and so the RNA chain is released, followed by the dissociation of the RNA polymerase enzyme, and this way the transcription terminates. Whereas in case of row-dependent termination, it requires the Rho protein. Rho protein is the ATP-dependent helicase enzyme that disrupts the DNA-RNA duplex. It binds with the growing RNA chain at RUT site. RUT stands for the Rho utilization site. Then it travels along the RNA chain in 5' to 3' direction with the help of energy. Energy is required in this step, which is provided by the ATP hydrolysis process, and it reaches to the DNA-RNA hybrid. When the RNA polymerase reaches the terminator, Rho protein catches up with it and releases the developing new RNA chain, which is followed by the dissociation of RNA polymerase enzyme. In this way, the transcription process terminates. So this was the gene transcription process. If you like the video, please like the video, click the share button, and please subscribe my YouTube video channel so that I can present more content like this. Thank you very much.